As we have seen in our previous two modules about the path of the oxygen from air bubble to the reaction site or inside the cell, we know that the solubility of the oxygen from the air bubble to the liquid medium, mean fermentation medium, is very critical. That is very fast process, but very critical. And other 5 to 7, and as we already discussed, that 5 and 7 steps are very slow. Here in this module, we will talk about the rate of oxygen transfer from the air bubble to the liquid phase can be expressed by this equation. You can see here that dCl over dt is equal to KLA into C star minus CL. In this equation, so this equation is very important equation in aeration and agitation point of view. Cl here is the concentration of the dissolved oxygen in fermentation broth, millimoles per liter. So that amount of the oxygen that is dissolved in the liquid, L designated by the liquid medium, mean fermentation medium. C is the concentration. So dCl is the change in the concentration of the oxygen with respect to time, and T is the time that is mostly expressed in hours. So dCl over dt is the change in oxygen concentration over the time period. That is actually the oxygen transfer rate, that is expressed in millimoles of oxygen per liter per hour. KL is, as you know that L is the liquid, so it is the mass transfer coefficient. Mass transfer coefficient. Then how is the transfer coefficient of the air or oxygen into the liquid? A is the gas over liquid interface area, that how much surface of the bubble, how much volume of the liquid interact to each other. Here the sparges that makes the air bubble size, as well as the air bubble size will be the less, there will be a more surface area. In the same case, so A is here the gas over liquid interface area per liquid volume, and C star is the saturation dissolved concentration, oxygen concentration, so at which there will be not more oxygen will be solubilized, because each and every media has its own capacity to dissolve the oxygen. So the saturation point is termed as C star. So this equation is very much important in order to discuss the oxygen transfer rate. So it is very difficult to calculate. In this equation, if we see that if we determine, it is very easy to calculate all other factors except KL, because KL is the mass transfer coefficient, and on the other hand, A is the area, interface area. So therefore the two terms are generally combined in a term that is known as KLA. So the KLA is known as the volumetric mass transfer coefficient. The unit of mass transfer coefficient, volumetric mass transfer coefficient, are the reciprocal with the time. So the volumetric mass transfer coefficient is used as the measure of aeration capacity in any fermentation process. The larger the KLA, the higher the aeration capacity of the system. So the KLU will depend upon the design of the fermenter, as well as the aeration and the agitation rate and the impeller design. So the KLA, volumetric mass transfer coefficient, depend upon fermenter design, impeller design, aeration and agitation rate. The KLA, which we called as volumetric mass transfer coefficient, that basically determine the oxygen transfer rate. So the oxygen transfer rate, which is termed as OTR, is the critical criterion in a fermentation. So the equation in the previous slide, you can see that it is affected by the KLa. The overall oxygen transfer rate is dependent on KLa and the dissolved oxygen concentration. So the dissolved oxygen concentration reflects the balance between the supply of the dissolved oxygen by the fermenter and the oxygen demand of the organism. So one is the dissolved oxygen and other is the demand of the cell. What will be the demand and what is the concentration of the oxygen present in the form of dissolved oxygen. So if the KLa is such that the oxygen demand of the organism can easily be met with the dissolved oxygen concentration will be greater than the C critical. So just to understand this phenomena of oxygen transfer rate, that is basically the driving force of the oxygen path. So if the ultimate need of the cell and then the rate of the oxygen transfer from the air bubble to the dissolved oxygen, and then actually if there will be no utilization of the oxygen, there will be no driving force of the solubilized. So the oxygen transfer rate is directly depend upon the volumetric mass transfer coefficient KLa and the utilization of any organism. So thus the KLa of the fermenter must be such that the optimum oxygen concentration for the product formation should be maintained. So if we maintain the KLa, that is basically met the need of that organism that required a sufficient amount, then we can run our fermentation process in a very suitable conditions to meet the requirement of any product formation, just to make our process economical.